So good afternoon, ladies and gentlemen. My name is Yulia Maximenko, and today I will be talking about microcapsules for luminescent tracking and controlled drug delivery. This work has been done in the University of Illinois and in collaboration with Max Planck Institute of Colloids and Interfaces and Saratov State University in Russia. So first I would like to discuss the motivation for our research, why we're studying what we're studying and why we're using these materials. Then I will discuss which particular materials we're using and for what reasons. And then I'll discuss some experimental results that we have so far. And in the end, we'll be able to talk about possible applications and further directions of research. So our motivation for research is to create a microcapsule with encapsulated drug for targeted drug delivery. And we would also like to be able to image those capsules, optically or electrically interact with them. The applications are straightforward. For example, for treatment of cancer or pulmonary tuberculosis, where medicine are usually very toxic and you wouldn't want them to affect your whole body or other healthy organs. So you would want all the medicine to be delivered directly to the particular tissue or particular cells, right? So we would have polymer shell with drug encapsulated inside, and we would want this capsule to be luminescent, right? Also, it is possible, if this content of this capsule is luminescent, it is possible to track the liquid flow after we break the capsule and release the drug at the point of interest. I think a very neat idea to create those capsules was using calcium carbonate microtemplates. They are porous, they are very cheap and easy to prepare. You can basically prepare them on your desk by just mixing two salts together, or you can use more sophisticated equipment to control their size better, to control their porosity better. They usually vary in size from sub-micron scale to 10 micron scale, and they are completely biocompatible, as you know, and they can be easily dissolved with acidic chemical treatment. So by simple incubation, we can incorporate drug into the calcium carbonate microparticle, as well as some luminescent agent that we would like to use to make our capsules luminescent. And it's a very good way to control size of your capsules, to control their spherical shape, because after you incorporate drugs in the pores, you encapsulate those particles with polymer of choice by simple adsorption on the surface. So to make our capsules luminescent, we are using silica nanoparticles. We use 3 nanometer silica nanoparticles. They are ultra bright and they have wide band emission in red-orange part of spectrum with the headband at about 650 nanometers. This is how the colloid usually looks like in isopropanol, so it's very bright and it's easily detectable either with your eye or with usual CCD camera. So 1 nanometer silicon nanoparticle looks like this. This is an example. So it's basically a piece of bulk silicon, but with pentagons on the surface instead of hexagons. Because of this shift, we observe shift in energy levels, and that's why they become optically active compared to optically dull bulk silicon. On the surface, silicon atoms are hydrogen passivated, and they form monohydrides. Dihydrides and trihydrides are also possible during HF processing, but by using hydrogen peroxide, we make sure that we don't have dihydride and trihydrides. So everywhere we have only silicon-hydrogen bond. So they're non-toxic, they're semiconducting, they are, the luminescence is very stable, and their bleaching is not irreversible. Even if they bleach, it happens only if UV is very harsh, about like 20 milliwatts or something. They can survive very well in neutral and acidic environment. They don't survive very well in basic environment because hydrogen gets easily removed by negative ions, and without hydrogen, nanoparticle is much less protected, so it gets easily dissolved then with hydrogen on top. For encapsulation, we're using polyethylenamine. This is a positively charged polyelectrolyte, which is forming a shell on the surface of calcium carbonates by simple adsorption, as I was saying. So polyethylenamine is known to form an incomplete layer, but it is a wetting agent for polyimide and polyimide shells, and it's a very good template for creating your own capsule, because then you can create multiple layers of polyimide if you need. You can vary different layers. You can control the thickness of the shell. You can control the permeability of the shell. But here we're just studying polyethylenamine capsules, and we study how well we can retain silica nanoparticles inside, and how well we can track those particles optically, and also it may be possible to electrically interact with this with capsules of polyethylenamine with nanoparticles inside, because nanoparticles are semiconductive, they are polarizable, they are also conducting in close spec formation. So it might be so our capsules would not be only optically active, but electrically active. PEI is also rather toxic, but some work is being done on minimizing its toxicity, by using degradable cross-linking and grafting agents. So now we can go into details about our experimental procedures. So first we prepare calcium carbonate templates by just mixing and homogenizing 2 solutions of salt. Then we prepare silica nanoparticles solution in liquid of choice using hexachloroplutonic acid catalyst. This procedure creates predominantly 3 nanometer nanoparticles with very good uniformity in size. So after this, we incubate our calcium carbonate templates in the silica nanoparticle solution, and you can see here bright luminescent calcium carbonate particles. Yeah, you could see on the previous slide just optical image of calcium carbonate, and here what we have now. So they're bright luminescent, and the average size is about 5 microns. Also I forgot to mention that PEI has distinctive blue-green luminescence under UV excitation. So we should be able to characterize our capsules by knowing the emission spectrum. So after coating calcium carbonate particles with silica nanoparticles, we incubate the complexes in PEI water solution. So here you can see that PEI is get absorbed on the surface, and you can see characteristic red luminescence of silica nanoparticles, as well as characteristic green-blue luminescence of PEI shell. So polyethylenamine presence was also confirmed by X-ray photoelectron spectroscopy. Here, for example, you can see a clear nitrogen peak at about 398 electron volts. So after coating our luminescent complexes with polyethylenamine, we dissolve calcium carbonate by just basic acidic treatment. We're using 1 molar HCl solution. So here you can see still luminescent capsules. The size of the typical structure is larger than 5 microns, but only because this is actually not one structure. It's just multiple structures clustered together, but not irreversibly clustered together. So if you look closer, you can see 6 or 5 capsules inside. So they're still luminescent, they're easily trackable, and we can see them in regular dark field microscopy. After we prepared those capsules and delivered them to our point of interest, then we can crack them with ultrasound or laser radiation. and release the content of the capsules into the surrounding environment. And it is also possible to track the liquid flow after that, because our nanoparticles are still luminescent, if they will be able to survive the surrounding environment. But for this, of course, you have to have very sensitive detection, probably single photon detection. Yes, so in conclusion, we prepared porous calcium carbonate spherical microparticles. We impregnated them with silicon nanoparticles and coated with polyethylenamine. And then we dissolved calcium carbonate templates with hydrochloric acid. So we observed that microcontainers display stable red luminescence, which is characteristic of silicon nanoparticles. And we see that we retain significant amount of silicon nanoparticles inside, even considering that polyethylenamine shell is not always completely continuous. Of course, we observed the drop in brightness, but it might be due to just simple volume dilution. And I will say later what can be done to improve this. So, as you could see, that optical tracking, optical detection of our microcontainers is very easily achieved, and we might be able to achieve tracking of liquid flow upon delivery of the microcontainer to the point of interest and breaking and releasing its content. So also one of the advantages of using silicon nanoparticles is that they are ultra small in size and they're spherical in shape, and they're expected to flush more easily than, for example, some larger, heavier, or needle-like nanomaterial objects. So also silicon nanoparticles are not toxic, and they form silicic acid in the body, which can already be found in the body, and this is a component of some basic medicines for common medical conditions, for example. So what can we do for future research? Of course, the whole point of this research is simultaneous impregnation of a drug and silicon nanoparticles in porous calcium carbonate templates. So you can pick a drug of your choice and you can impregnate simultaneously our microparticle templates, and then just repeat the whole procedure covered with polyethylenamine, and then release it when you would need to do this. So as I was also saying, polyethylenamine is a wetting agent for polyamides and polyamides, and you can control very well thickness of your shell by just adding different polyamides on top of your containers before you dissolve calcium carbonate, and you can control permeability of the shell. And also that would help to retain almost 100% of nanoparticles inside. And another trick that you can do, you can incorporate silicon nanoparticles between layers. So you can incubate complexes in any stage in silicon nanoparticle solution and then coat it once again with some other layer. And you can do it with as many times as you would like, thus significantly increasing luminescence of your complexes. Also, one application would be using magnetic silicon nanoparticles instead of just simple silicon nanoparticles, and we can make them magnetic by combining silicon nanoparticle and some magnetic ion, for example, erbium or iron. It's a very simple procedure, and we proved that those ions form a stable complex with the silicon nanoparticle. This is an example of 1 nanometer silicon nanoparticle with the ion residing just outside, which is a more stable combination than this one, but this is also can be possible. With nanoparticles of larger sizes, it is possible for a nanoparticle to combine at the same time with several magnetic ions. So that would make our nanoparticles magnetically active, electrically active, and optically active, and you can do the same procedure and make your capsules luminescent, and then you can control, you can add additional means of control by magnetic field, for example, for your experiment. And I think that's it, and thank you very much for your attention.